Hello, dear students. Hope all of you are fine. Students, as you know, we are in lockdown and we are stuck in our homes due to the reason you all are aware about. And the and the situation demands also. Students, in such situation, it is very likely that uh, you may feel anxiety and stress. uh, the best way to keep that stress and anxiety away from you is to keep yourself busy, and on priority basis keep yourself busy with your studies, and spend your time with your family, your parents, and take a very good care of yourself and your family. Stay safe. So we are so let us move to the our topic, and hopefully this will be our last lecture on phylum Porifera. Okay, so students, we are discussing today this canal system. Okay, it is the distinguishing feature of the sponge. Okay, and actually, you have studied in the last lecture that the sponge body is perforated by numerous apertures. Okay, and these apertures are for ingress, that is inward movement, and egress, outward movement of water inside the body. This water which enters into the body flows through certain system of spaces and channels, collectively forming the canal system. Okay, there are certain system of spaces inside the body through which this water flows, and this forms the canal system in the body of sponge. uh, it plays a very vital role in the physiology of these animals. All the exchange which are done between this animal and environment is maintained by this water current. Food and oxygen and that are present in that water current moves into the body, and wastes and reproductive bodies, that means those gametes and asexual structures such as gamules, move to the outside environment uh, with water. So this plays a very important role in the physiology of these animals. Actually, the current by which the water flows into the body of these animals is caused by these collar cells or choanocytes, which are flagellated cells, and I have already told you that these cells are characteristic cells of this phylum. Students, arrangement and complexity varies in these animals as far as this canal system is concerned, and on the basis of arrangement and complexity of this canal system, the canal system is of three types: ascon, sycon, and leucon. Let us move to the first type, that is ascon. Ascon is the simplest type of canal system, and sponges which have this type of canal system are called as asconoid sponges, and the best example in this is leucon solonia. Okay, the course of the water which the water follows in this type of canal system is that water enters into the body through ostia, and then it moves directly into the spongocele, that central cavity, and from spongocele it moves to the outside through this osculum. uh, let me show you this in this diagram. Look, students, these are dermal ostia. Water enters through dermal ostia, and due to these flagellated cells, this water current is caused, and water moves from this ostia into the spongocele, this central cavity, and from central cavity it moves out through this osculum. This exit pore is called as osculum. Okay, let us move to the second type of canal system, which is uh, which is this Cycon type. It is more complex type of canal system, and the spawns having this type of canal system are cyconide spawns. Examples include Skypha and Grantia. Theoretically, it is derived from the folding horizontally of this esconide type. Okay, the course of the water in this type of canal system is water enters through dermal ostia and it moves into the incurrent canals. Okay, the first type of chambers. These incurrent canals are non-flagellated. From non-flagellated, these incurrent canals, it moves into the flagellated chambers called as radial canals, and connection between these two is prosopiles. Okay, and from radial canals, it moves into the spongocoel, which these radial canals open into spongocoel through apopiles, and from spongocoel it moves to the outside through this osculum. Okay, let us see this diagram. Look here. The water enters through this dermal ostia. Okay, and it enters into through this dermal ostia, it enters into these chambers, these non-flagellated chambers, and these non-flagellated chambers are called as incurrent canals. And from these incurrent canals, water moves into these flagellated chambers. Okay, flagellated chambers, which are called as radial canal, and from radial canal it moves into this spongocoel. Okay, and from spongocoel to the outside. Okay, this was the second type of canal system, which is Cycon type of canal system. And now let us move to the most complex and third type of canal system, that is Leucon type of canal system. Further folding of body wall of this Cycon type gives rise to Leucon type. Okay, and these spawns are called as leuconide spawns, and best example in this category is spongilla, which is also the freshwater sponge. And the course of water is that water enters into the body through dermal ostia in all three cases, and from and there the water moves into incurrent canals. From incurrent canals it moves through prosopiles, it moves into flagellated chambers. Okay, another type of spaces. And from and through apopiles, from flagellated chambers it moves into the excurrent. These canals are extra in this type of, and these excurrent canals are developed as the result of shrinkage and division of spongocoel. Okay, in this type of canal system you may notice that the this central cavity, that is spongocoel, is highly reduced, and that cavity is reduced, and it forms these excurrent canals, and from excurrent canals water moves to the outside through osculum. So let us see this diagram. The water enters into these incurrent canals through dermal ostia. Okay, and from dermal ostia it enters into these flagellated chambers. Okay, and from flagellated chambers it moves into this excurrent canal. Okay, these excurrent canals, these excurrent canals are actually formed due to the shrinkage of this spongocoel. You may notice here that this spongocoel is highly reduced, and from these these radial these excurrent canals, the water moves into this shrinked or reduced this spongocoel, and from spongocoel to the outside. So this was all about this canal system, a characteristic feature of these spawns. And as I told you earlier, that it plays a very important role in the physiology of these animals. And water takes in oxygen and food, and then brings out those wastes and reproductive bodies, such as gametes and asexual those asexual gamules, etc. And with this, or phylum Porifera is completed, and hopefully we will start the next phylum in the next lecture. Okay, thank you.